Hello, I am Hervé Ménager from the Institut Pasteur in Paris, an EDAM community co-lead.
Hi, I am Matusz Kalas from the University of Bergen in Norway. I am a community co-lead and a maintainer of EDAM. This presentation is on behalf of the global community of EDAM contributors. And we thank the Bio-Ontologies COSI for the opportunity to present EDAM, an ontology of data analysis and management, at ISMB. The main links for starting engaging with the EDAM ontology are displayed at the top of this slide. EDAM is part of the portfolio of services provided by Elixia. Maintainers of EDAM are supported by the French Institute of Bioinformatics, which constitutes the French node of Elixia, and by Elixia Norway. Before we dive into EDAM, let me show one historical example. BioCatalog has been a great registry of live scientific web services. This screenshot was taken back in 2014 from a version at that time, when trying to find services that perform sequence similarity search in protein domain families. Using a search phrase, sequence similarity search protein domains, in a full-text search, unfortunately returned no hits, although there were services doing exactly that. One could ask, what are the suitable search terms? Wouldn't it be nice to have an option to filter by some categories, like in an online shop? This is one of the scenarios that motivated the development of the EDAM ontology. So what is EDAM? EDAM is a domain ontology of data analysis and research data management, consisting of 4 sections: topic, operation, data, and format. The topic section includes scientific and application domains, data acquisition techniques, and other key notions related to a research workflow as a whole. Examples are paleogenomics or electron cryotomography. Operation, on the other hand, comprises concrete actions that can be performed with data, usually producing some output data based on the consumed input. Examples of operations are molecular docking or image segmentation. Data includes concepts representing types of data, independently of which formats the data could be serialized in. Both complex, structured types of data are included, for example, sequence assembly, as well as simple, atomic data such as gene ID. Format includes concrete, specified and documented formats of data. For example, FASTQ, or the Systems Biology Markup Language, SBML. These are supplemented by auxiliary concepts, grouping related formats together. The structure of EDAM is quite simple, with a couple of relations between EDAM concepts, in addition to the main hierarchy. An operation may have a topic, and inputs and outputs, which are data. A data concept may also have a topic. A format is format of some data. And types of identifiers, forming a distinct subsection of data, can be identifiers of some data or of other entities. With respect to the usage areas, EDAM has been primarily designed for finding tools, workflows, learning materials, standards, and other digital objects. Or to see it from the opposite side, for annotating digital research objects related to data analysis and data lifecycle, in order to make them better searchable. A special case is recording provenance metadata, where EDAM can help documenting, for example, the performed processing steps or the data acquisition technology, making the data more understandable, searchable, and comparable. However, just like making cities friendly for users of wheelchairs makes them better for everyone, an ontology for annotating and finding tools and other research objects has proven itself useful in additional usage scenarios. Well-annotated tools and data are easier to integrate. EDAM annotations help filtering suitable input data, and the matching of inputs, outputs, and operations can even help in semi-automated workflow composition. As any domain ontology, EDAM can be used in text mining. Last but not least, EDAM has proven itself helpful for scientists writing about their data processing and analysis and needing to find the correct terms, such as in the method section of an article or in workflow documentation. EDAM aims at being usable together with other ontologies, interoperable. It is maintained as a rather typical oboe-in-owl ontology, with a touch of additional standard semantic web vocabularies. EDAM is distributed under the Creative Commons share-alike license, which is a so-called weak copyleft. This means that modifications must be distributed under compatible terms. However, it allows inclusion in other work distributed under any license, without restrictions. EDAM is linked to various upper ontologies, including BFO, GFO, Biotop, and Dolce. This should allow diverse reuse options in scenarios requiring a common upper ontology. In addition, EDAM links to other ontologies, knowledge bases, and vocabularies in situations of an overlap, or when another ontology or knowledge base provides additional levels of detail. EDAM extensions enable specialized scientific communities to develop EDAM towards their needs. One major extension is EDAM bioimaging, dedicated to bioimage informatics and image analysis. EDAM bioimaging has been developed by the community of imaging and image analysis experts, with essential contributors from 12 European countries and a couple of countries outside of Europe. The image analysis community contributed also the coverage of machine learning concepts in EDAM, which are now part of EDAM bioimaging. REMBI, a proposal for standardization of bioimage metadata, suggests the usability of EDAM bioimaging for various parts of the provenance metadata for biological images. Details are listed in the supplementary material of the REMBI article. Since 2021, the EDAM Geo community has been forming and extending EDAM towards geolocated data and spatial sciences. Examples include public, global, and planetary health, with application domains such as epidemiology or toxicology, environmental sciences, climate and pollution, or highly interdisciplinary Earth system modeling. This illustration shows topics added among biosciences, such as geobiology, and additional high-level domains, including geosciences. EDAM Geo covers also geographical data and geoinformatics data formats. Both EDAM Geo and EDAM Bioimaging will be iteratively merged into the main EDAM to smoothly serve the needs of the cutting-edge interdisciplinary research applications. If you're interested in contributing to this work, or have an application that would benefit from EDAM Geo, please connect via GitHub, Twitter, or Web Protege. The EDAM Geo work has so far been following the needs of the CWL, Galaxy, and EOS Nordic communities.
EDAM originates from the bioinformatics open source community, and its contribution model is heavily influenced by this origin. This model is community-based, meaning the EDAM community relies on contributions from experts who help correct and enrich the ontology over time. To facilitate this process, we provide a set of tools and workflows. Some of these tools support the exploration and assessment of the EDAM concepts for users and contributors. Others perform the validation of the ontology for EDAM curators and maintainers. These tools include EDAM Browser, a visualization interface that lets users explore EDAM in a simplified way. It includes multiple custom functions adapted to the needs of users, such as the visualization of the usage of concepts to understand them better and assess their use. or the possibility to directly submit new suggestions to GitHub using the graphical user interface. EDAM validation is performed by a set of workflows that run quality control tests, in addition to incorporating standard tools such as ELK and Robot. We developed our custom tool that runs some additional tests at both the syntactic and semantic levels. To illustrate the practical use of EDAM, let me give you a few examples, starting with BioTools. BioTools is a registry including over 25,000 software tools and services. EDAM allows the formal description of tools, starting with their area of focus, using topics such as functional genomics in this example, but also the operation they perform, here prediction and recognition, and the nature and format of the data consumed and produced by the software. In this example, consuming FASTA sequences as input data. EDAM is used also by TESS, the Elixir training portal, where it is used to categorize training materials and events. Another example is its use in fair sharing, where EDAM, together with various other ontologies, is used to categorize information standards, policies, and databases. EDAM annotations are used in diverse other resources to help searching for tools and other digital objects. For example, in the Galaxy Workbench, the XPC portal of the Swiss Institute of Bioinformatics, or the BIII registry of bioimage analysis resources. Additionally, EDAM is already at the heart of several research activities in computer science. For instance, some work aimed at accelerating and facilitating development of bioinformatics workflows with composition and recommendation support tools. Other works aim at the verification of datasets. Based on provenance traces, it is possible to annotate data with semantic annotations associated to analysis tools. The analyzed datasets can thus be automatically annotated with EDAM for better indexing and sharing. As a conclusion, EDAM is an ontology widely used to describe data, methods, and other resources in life sciences and beyond. It is deployed in diverse large-scale registries and databases and parts of various standards. EDAM is developed in a participatory manner, assisted by a set of tools, and we warmly welcome new contributors.